So a timely topic given the situation we are currently living in. Let's talk a little bit about the immune system as it relates to its anatomy. So when we look at the system in terms of the cytological responses that we're going to see, we have to start looking at what differential cells we'll see. And most of the differential cells that we're going to see is based off of what's referred to as a CD factoring that comes into play in the notes packets. I've given you various types of CD factors. These CD factors are the differential factors or the cluster differential factors that are going to be linked to the initiation of the immunological responses that we see within the cells. We break this up into distinct parts. In terms of the lymphocytes, we have three distinct types of lymphocytes. We have what we refer to as the NK or the natural killing cells. Then we have the thymus lymphocytes or the T lymphocytes or the T cells. These are the cells that will be maturing within the thymus gland. We have three distinct types of these T cells based off of their activation sequences. We have memory T cells. These are T cells that are remaining after an active pathogen response. We have helper T cells. Helper T cells are going to initiate the cytological responses via cytokine or hormone releasing factors, which include IL-1 and IL-2. These helper T cells can also function as sequester T cells. These are T cells that will limit the total amount of response that we see going on within an immune response. And then we have the killer or the cytotoxic T cells. These are the T cells that are going to be actively involved with the immune response in terms of digestion and absorption of the pathogenic entity. The last are the B cells. The B cells are the bone-derived lymphocytes, or the B lymphocytes, or the B cells. Just like with T cells, we have different types of B cells. We have memory B cells. These are B cells that have embedded immunoglobulins into the membrane that allow for binding onto known pathogenic antigens. We have clonal B cells. These are B cells that are undergoing replication upon stimulation of IL-1 and IL-2. And then we have the plasmal B cells. The plasmal B cells are the B cells that are actively involved with the response to the pathogen in terms of production and release of antibodies. Along with the lymphocytes, we have macrophages. Macrophages are going to be cells that will ingest. We have the granulocytes, which include the eosinophils, the basophils, the neutrophils, and then we have the thrombocytes or the megakaryocytes, which are going to be involved with the clotting of the blood in response to vascular injury. The eosinophils, basophils, and neutrophils, as well as the megakaryocytes and macrophages, are going to come from the same cell lineage that gave rise to the erythrocytes, whereas the lymphocytes are going to come from a differential stem cell. And so if we look at the growth that we see within the cells, and we'll see a video here in a second, we have what we refer to as the myeloid stem cells. The myeloid stem cells will give rise to the erythrocytes, the thrombocytes, as well as the granulocytes, and the macrophages, which include the monocytes indicated here in the flowchart. All of these cells are going to be, in terms of growth, regulated based off of differential hormonal growth. The hormonal growths include various types of growth signals, which include IL-1 and IL-2, thrombopoietin, TPO, which is going to initiate cell growth just the same way that EPO regulated the growth that we see with the erythrocytes. In this, we can differentiate their responsiveness, where the myeloid-derived stem cells are going to give rise to the innate or the non-specific immune cells. These innate non-specific immune cells, even though they have a hormonal regulation involved with their growth factoring, they are not hormonally regulated in terms of growth. They will naturally have a cell cycle, cell division processes where they tend to have very short lifespans. Whereas the lymphoid tend to be specific and are involved with the acquired immune responses. These tend to be very long-lived cells and are heavily regulated based off of hormonal regulation and development.
Our body is constantly making new blood cells. Billions of white blood cells, red blood cells, and platelets are produced every day to replace older cells that die, either during normal processes or because of illness. This process by which new blood cells are created is called hematopoiesis. Blood cells in the body, including the white cells, are made in the bone marrow, which is the soft, spongy material in the center of the bones. Blood cells are created from a group of master cells called stem cells, which also live within the bone marrow. These stem cells have the ability to divide and produce all the different types of blood cells. The white blood cells are an important part of our body's immune system. Granulocytes are a special group of white blood cells that play an extremely important role in protecting the body against infections. Neutrophils are a special class of granulocytes that are able to chase, engulf, and digest the bacteria that can cause infections. Neutrophil development in the bone marrow takes approximately 10 days. Mature neutrophils are released into the blood where they survive for only 3 to 6 hours. Because mature neutrophils are so short-lived, the bone marrow constantly produces large numbers of neutrophils to meet the body's needs. Our body makes natural proteins that can signal the stem cell to start making cells that will eventually develop as neutrophils. One of the important proteins in this process is called granulocyte colony stimulating factor, or GCSF. The GCSF protein can work on the cells in the bone marrow that eventually become neutrophils and signal that they should start becoming neutrophils. The entire process by which stem cells develop into mature neutrophils can take about 10 days and includes 2 phases. During the first phase, the stem cells and their offspring can divide as they develop and mature. This step takes about 6 days. Then, over another 4 to 5 days, the cells mature further. However, the cells can no longer divide. The mature neutrophils are finally released into the blood, where they can fight bacteria and protect the body from infection.
So let's take a look at these cells that we just saw developing out of the video, and we'll look at the myelocyte or the granulocytes, which are the first responders. These include the basophils. The basophils will emigrate into any tissue that we happen to have infection injury. These are the least numerous of the granulocytes. They will exocytotically release granules of heparin, histamine, and serotonin. These chemical signals, these hormones, will initiate the inflammatory responses and are involved with acute reactions within tissues. The histamine and serotonin that is released from the basophils, and specifically from basophil-initiated dendritic cells, will be involved with the allergic reactions that we see. Most of the anti-allergy medicines that are given are there to regulate the actions of the basophils, as well as the mast cells and dendrites, by limiting the amount of histamine and serotonin that are being released, as well as a number of other cell signals, particularly chemoattractants, which are going to drive cells into the areas that are going to be involved with immune response. The next one is a little bit more numerous than we see with the basophils, and these are the eosinophils. The eosinophils will emigrate into interstitial fluids based off of chemoattractant signals. Just like with the basophils, they will be involved with the acute responses. They will release an enzyme known as histaminase. Histaminase will digest histamines, thereby limiting the total amount of histamine response that we see, which is important in terms of regulating the amount of inflammatory response that takes place to any type of infection or injury. We see them highly involved with any type of parasitic infection. They are involved with a phagocytotic process. When we see an antigen-labeled marker, that means that the B cell has indicated this happens to be a foreign pathogen. The last one that we're going to see is the neutrophil. The neutrophil is the most numerous, as we just saw in the video. These are going to move to areas based off of chemotaxic signals, chemoattractant signals, and will be involved with any type of bacterial or viral infection. They will initiate a chemotoxic response, thereby releasing their granules into the interstitial areas that will digest the pathogen through either lysozyme action or through release of oxidants and defensins. Defensins are secondary complementary proteins which will instigate further responses to immune activation. The last of the myelocytes are the monocytes. The monocytes are not a granulocyte. They are an actual phagocytotic cell. They are going to be late arriving to any type of area of infection or area of damage. Their principal action is to undergo phagocytotic ingestion of any of the digested pathogenic entity or any of the tissue that has been damaged and is in need of repair. All of the myelocytes, the granulocytes, the monocytes, are anaerobic obligates. They do not want to live in an oxidative environment. They do not want to have large amounts of oxygen in their environment. These are the cells that we see in pustulates around injuries. If you happen to be treating any type of wound that is pussing, the worst thing you can do is put any type of oxidizing agent, such as hydrogen peroxide, onto that area, as it will alleviate the anaerobic nature of the area and thereby diminish the quality of work that these cells can take. The best thing you can do if you happen to have or be dealing with any type of pustulating injury is to simply cover the area so as to minimize any secondary infection that can take place and allow the pus to accumulate around the area. You can go ahead and clean away some of the excessive pus. However, you want to allow it to continue to pus because that's the indication that these cells are functioning correctly. The other type of immune cells are the lymphocytes. The lymphocytes are going to be specialized immune cells that will reside predominantly within the lymphatics. About 70 to 80% of all fetally developed lymphocytes will become T lymphocytes or T cells. The remaining 20 to 30% will become B cells. The T cells will undergo secondary differentiation and maturation within the thymus. The T cells, we want them to be able to recognize the self-antigen but not to become active to the self-antigen. The B cells, we do not want to recognize self-antigen whatsoever. If a B cell recognizes the antigen, means it's able to lock onto the antigen, it will produce antibodies or immunoglobulins to that antigen. It is the B cells and the T cells that are involved with immunocompetence, that is the ability to respond to a given pathogen. If we happen to become immunocompromised, that is where we lose the lymphocyte activation to distinct pathogenic entities. Immunocompromisation will occur with things like leukemia or with infection with the HI virus, the human immunodeficiency virus, or HIV. So if we look at the maturation of the lymphocytes, what we end up having is the stem cells being derived from the red bone marrow within the trabecular region of the long bones. 70 to 80% of these cells will be released into circulation and head to the thymus. In the thymus, they will undergo secondary processing, leading to what's referred to as the undifferentiated lymphocytes. These are lymphocytes that will develop into T cells and are simply going to be waiting for activation to a pathogen in order to become active and thereby become either helper or killer. The remaining 20 to 30% will continue to mature within the bone marrow based off of their maturation sequence. If they are able to not recognize the self, the B cells will then be released into lymphatic circulation, and for the most part, they will remain within the medullary cortex of the lymphocyte and within the germinal cell region of the lymph node. The T cells will mature within the thymus and then be released. The T cell differentiation will slow down as we go into puberty simply because of growth factoring that will impact the thymus's ability to continue to allow for maturation of the T cells. The T cells will spend most of their time in circulation. We will not see as many of them housed within the lymph node as we see of the B cells. So the last thing we need to talk about here in terms of the cytological responses is the maturation of the system. The system itself is an ever-maturing system based off of access to all of the various pathogens that we're going to be interacting with. And so we can say that the system never truly will fully mature. The system itself doesn't mature beyond the ability to self-recognize until after birth. Early in development, we do get some exchange of immunoglobulins from the mother to be via placental exchange, but most of what's going on in fetal development is simply the learning of the self-antigen. The first stages of total maturation occurs during infancy in neonatal care with exchange and reception of antibodies from the mother via breast milk and exposure to maternal bacterium during birthing and from bacterium on the surface of the skin around the nipple during breastfeeding itself. So the system itself is never going to fully mature based off of all of the various interactions that we have going on, where full maturation will take place at the time of death of the individual. However, even though we're saying it's always constantly maturing, we aren't generating as many new cells as we think we might be generating late in life, particularly with the T cell maturations as the thymus undergoes atrophy during pupil development, limiting the total amount of T cells that can be fully developed post-pubertal development. Most of the maturation that we see going on is in the immunocompetence responses that we see cytologically to the various pathogens that we'll be interacting with throughout our lifespan.